1945, the group of scientists, so they have, through electron micrograph of the cells, they could find some lace shaped network of strands in the cytoplasm. And then Pellad and Porter, they mentioned key characteristics of endoplasmic reticulum and ribosomes. So that means it is like a evolution, like how the discovery of endoplasmic reticulum has happened. Okay. Then in 1966, George Pellad, he generated a transmission electron micrograph illustrating the endoplasmic reticulum, which is studied by granules, that is ribosomes. So whenever endoplasmic reticulum is attached to ribosomes, then it is called as rough endoplasmic reticulum. So they have given a structure of endoplasmic reticulum, which was taken under transmission electron microscopy. Okay. And next in 1974, the same scientist, he continuously worked on that, and he described the structure of endoplasmic reticulum and Golgi complex. So both he has given. So this, the third image, is the scientist who have given this. Now this endoplasmic reticulum is seen in almost all the cells except red blood cells of the mammals, right? So in animal cells, this endoplasmic reticulum membrane, it is almost half of the total membranous system of the cells. So it is, as it is folding very intensely, we cannot see how much membrane is present. But due to this foldings, its surface area will get increased. Okay, so if we take a eukaryotic cell or animal cell, so if out of the total content of the membrane which is present in the cell, more than half of that is contributed by this endoplasmic reticulum membrane. And you cannot see this endoplasmic reticulum in prokaryotic cells. And this is, the endoplasmic reticulum is enclosed in a continuous membrane and is the largest organelle of the eukaryotic cell. So that's why, because of foldings, we cannot see how large it is, but it is the largest one. And its membrane may account, so more than half of the cell membranes. And the space which is enclosed by this membrane, okay, so within that, that is called lumen or cisternal space. So this contributes about 10% of the total cell volume, right? So then the endoplasmic reticulum is an extensive system. It consists of tubules which are branched and some flat discs or sacs which are called cisternae. Okay, this will get extended throughout the cytoplasm, that is from plasma membrane to nuclear membrane. You can see these foldings of endoplasmic reticulum. So it is continuously extended throughout the cytoplasm. So how this endoplasmic reticulum has originated? So there are some concepts which were proposed previously, but according to current concepts, so it's this membrane was erased, or biogenesis means birth, right? So how it has origin is through a multi-step process that involves the synthesis of a basic membrane made up of lipids and intrinsic proteins. So yesterday we have discussed about membranes and proteins which are present. Then the addition of other components such as enzymes, sugars, lipids has occurred in a sequential manner. And the insertion of proteins is independent of that of lipids. That means it is not that for this lipid, this protein has to be there. There is no dependence of proteins on the lipids. So both are independent. And some proteins of endoplasmic reticulum are formed by the ribosomes in the cytosol and then become inserted in the membrane. So we have ribosomes attached to the endoplasmic reticulum and also which are free in the cytoplasm. So actually the main role of ribosomes is protein synthesis. So whatever the proteins which are synthesized in the ribosomes present in the cytosol, that got inserted into the membrane of the endoplasmic reticulum. Okay. So there are some microsomes which are present in the endoplasmic reticulum, and how can we remove that? Okay, so here you can see the endoplasmic reticulum, which are like attached to the, sorry, which are linked to ribosomes. These are rough endoplasmic reticulum and these are smooth, as there are no there are no ribosomes. So here, first this endoplasmic reticulum has to be homogenized. That means we have to grind it. Okay. Then the rough and smooth microsomes. So those small round shaped vesicles. So that will be separated. And these can be done by the sucrose concentration gradient. So usually we do gradient electrophoresis or gradient centrifugation. So where like we are giving different concentrations of sucrose. Okay, sucrose gradient centrifugation, where bottom it will have more concentration and then a little bit less concentration. Like that we will change the gradient and we will centrifuge. So different components also we can use. Suppose if you have DNA, RNA, protein, like that, if you want to separate these three, you can add these three samples in the tube and we can centrifuge. And these three will have different gradient, different molecular weight. Therefore, after centrifugation, we can see their positions at different concentrations of the sucrose, wherever the molecular weight matches. Okay, so now the sample has been added and centrifugation was performed. Now smooth microsomes or the smooth portions of the endoplasmic reticulum, they will have low density because there are no ribosomes are there on that. So they will appear at the top where the low sucrose concentration is present. Whereas the rough microsomes have more density, therefore they will be at the bottom where the high sucrose concentration is present. Okay, so these are the regions of the endoplasmic reticulum. Okay, so yeah, so this was a study how Peled has performed, but that is the concept. No need to go in depth. Now we will discuss about the structure of endoplasmic reticulum. So endoplasmic reticulum extends from nuclear membrane at one end and joins the plasma membrane at the other end. So it is throughout the cytoplasm it extends. It never opens into the cytoplasm. That is, it is closed by the membrane, but it is extending from this end to that end. And it is continuous with other organelles such as Golgi apparatus and mitochondria. And all these structures like sacs, tubules are held by cytoskeleton. That means if you see the cytoplasm is fluid, and usually the organelles also will be very flexible. They won't be rigid. Because though membrane is present, they will be usually flexible. But whenever the sacs and tubules are present in the fluid, that has to be maintained in proper position. And that position was maintained by this cytoskeleton. And the endoplasmic reticulum remains bound by this phospholipid membrane. Okay. And inside the membrane, have a fluid filled cavity that is called as cisternal space or lumen. This lumen is in continuous with the perinuclear space. So now what kind of enzymes are present in this endoplasmic reticulum membrane? Okay. And we also have stereases, NADH cytochrome reductase, glucose 6 phosphatase, glycosyl transferase, and magnesium activated ATPase. So these are the enzymes. And along with these enzymes, they also contain triglycerides, phospholipids, and cholesterol. Okay. So in electron micrographic study, the cross section of endoplasmic reticulum appeared to be two parallel membranes separated by a light space, which is called cisternae. So here is the image, how it looks like. So if you see, this is the external medium. And this is the inside of the cell. So where you have nucleus and the space around this like nuclear membrane. So that is perinuclear space. So this cisternal space is present here. So this endoplasmic reticulum is in continuous with Golgi apparatus, which is present here. So this is along with the nucleus also. And these are smooth endoplasmic reticulum, SER, and RER is rough endoplasmic reticulum. So here you can see the clear structure, how it looks like. So this is a nucleus to which the endoplasmic reticulum is continuous till cell membrane. So it is distributed all over the cytoplasm. Now, what are the components of this endoplasmic reticulum? So based on the morphology, there are three types of structures present: cisternae, tubules, and perinuclear space. So cisternae are nothing but they're long, flattened structures. They're not branched, okay? And they are arranged in parallel bundles, okay, one after the other. And each tubule is having 40 to 50 micrometer thickness, okay? And second one is vesicles. So vesicles are, as we have discussed in the first class, vesicles will be there. They are round shaped or ovoid structures, and they are surrounded by the unit membrane, which are isolated in cytoplasm. That means they are surrounded by the membranes so that there will be distinguishing. Okay. Next are tubules. Tubules have irregular shape and irregular branching also, where the active biosynthesis of glycerides, okay, triglycerides will take place. And in this you can see these tubules in epithelial cells of retina and also involved in the metabolism of vitamin A, which is retinol. Now, along this endoplasmic reticulum, there are other three components which are bounded by thick membranes, and they are perinuclear space, that is the space between endoplasmic reticulum and nucleus. Okay, so yeah, so this is how the cisternae looks like. You can call, okay, you can see here. Then vesicles and tubules. So how they look. Now the main functions of endoplasmic reticulum are mainly synthesis and translocation of secretory proteins across the rough endoplasmic reticulum. So rough endoplasmic reticulum means where the ribosomes are present. So these ribosomes will produce proteins and they will, like, that has to be transported. So the synthesis takes place here and the translocation will be taken place by the endoplasmic reticulum and the packing will be done by the Golgi apparatus. And integration of proteins into the plasma membrane. So in the plasma membrane, we have different types of proteins like transmembrane proteins, peripheral proteins, lipid anchored proteins. So like that. So that insertion is done by this endoplasmic reticulum. Then folding and modification of proteins in the rough endoplasmic reticulum lumen, and synthesis of phospholipids and steroids in the cytosolic side of the smooth endoplasmic reticulum. So these both phospholipids and steroids, which are important in plasma membrane structure. Then storage of calcium ion in smooth endoplasmic reticulum lumen, and their regulation, their release into the cytosol is regulated by endoplasmic reticulum. Then some carbohydrate metabolism and detoxification of drugs also will be taken care by this endoplasmic reticulum. Please mute yourself. Yeah. So what are the different types of endoplasmic reticulum? So there are two types: smooth endoplasmic reticulum and rough endoplasmic reticulum, based on absence or presence of ribosomes. So they are called as granules here. So you can also call the smooth endoplasmic reticulum as agranular. That means the granules are absent here. So therefore the ribosomes are absent and they do not attach to the surface, hence they are called agranular or smooth endoplasmic reticulum. And those endoplasmic reticulum which have ribosomes on its surface, they are called granular or rough endoplasmic reticulum. Like they can change. That means it is not that always the smooth endoplasmic reticulum has to remain smooth endoplasmic reticulum only. No, it can become rough endoplasmic reticulum also by attaching to the ribosomes, or rough endoplasmic reticulum can convert into smooth endoplasmic reticulum. So according to the need, if the cell requires more protein synthesis, then this conversion can happen. So this is how they look. Those which are granules are present, that is rough endoplasmic reticulum, and where the granules are absent, that is smooth endoplasmic reticulum. So first we will discuss about smooth endoplasmic reticulum. It is highly curved and tubular structure, and it doesn't have ribosomes. The tubules are 500 to 1000 angstrom long, and there are irregular lattice. And these are present mainly in the cells which are connected with steroid and lipid biosynthesis and carbohydrate metabolism. That means here there is no role of ribosomes, right? If there is protein synthesis is required, then ribosomes play important role. As here the steroid, lipid and carbohydrate synthesis or metabolism is taking place, so mostly we say smooth endoplasmic reticulum here. And different amount of smooth endoplasmic reticulum also we can see. That too depends on the nature and function. So these are mainly involved in lipids synthesis, that is mainly cholesterol and phospholipids. So which has to be inserted into the new cellular membrane. That means whenever the cells divide, they require plasma membrane. So whenever the plasma membrane is synthesized, they require phospholipids, cholesterol, all these things. So that requirement is fulfilled by this smooth endoplasmic reticulum. In some cells, this is involved in the synthesis of steroid hormones like cholesterol. So cholesterol as a part of plasma membrane, it is very less. The role is very little. Okay, the amount of cholesterol present in the membrane is also little. But apart from that, cholesterol has many other functions in the cell. So that is fulfilled by this type of endoplasmic reticulum. And in cells of liver, it contributes to detoxification of drugs and harmful chemicals. So we all know, like whatever the drugs or whatever it take, that first goes to the liver, and the detoxification of that takes place in liver. So in that, this endoplasmic reticulum will assist. And in, yeah, any doubts? Okay. So in muscles, it is called as sarcoplasmic reticulum. Okay. So it's a specialized type of smooth endoplasmic reticulum which regulates calcium ion concentration in the cytoplasm of striated muscle cells, so that the contraction of muscles is regulated. Now the functions of the smooth endoplasmic reticulum. So as I told you, now whatever we have discussed, all these are the functions like protein transport, steroid, carbohydrate metabolism, drug detoxification, and synthesis of cholesterol and phospholipids, which are major components of plasma and internal membranes. So these are the overall functions of the smooth endoplasmic reticulum. So if we see one by one, if any points are missed, we will cover. So first is transport of proteins. where the smooth endoplasmic reticulum, though they are not involved in the synthesis of proteins, they are route for the transport of metabolites such as proteins through the formation of vesicles. Okay, so this smooth endoplasmic reticulum transports the proteins which are formed in the rough endoplasmic reticulum, which have ribosomes. So that will be transferred to the Golgi apparatus where the packing will be taken place, and that is sent to the different parts of the cells. Okay. Second is steroid metabolism. Here also the smooth endoplasmic reticulum is involved in steroid metabolism, where we have steroid derivatives like cholesterol and the steroid hormones, then bile acids will be synthesized. And the next is release, uptake and storage of calcium ions. So this smooth endoplasmic reticulum, it attaches to the receptors of calcium binding proteins. Okay. So whenever the proteins goes and binds to the calcium, that will come and bind to these receptors. So the calcium which is stored in the striated muscle cells, so that is contributed by the smooth endoplasmic reticulum, and that calcium helps in muscle contraction and relaxation. So I told you, like, it is also called as a sarcoplasmic reticulum in muscles. So from the cytosol or cytoplasm, the calcium is taken up inside the muscle by calcium ATPase pump, and that intracellular calcium is helpful for the muscle contraction and relaxation. Okay, and that too in response to the external signals. If there is any like if you want to run, so what happens? All your muscles starts functioning. So that means it requires more calcium. So that is one kind of the signal. Okay. And the next is lipid metabolism. So the synthesis of fatty acids, glycerols, steroids, like cholesterol, everything takes place here. And there are other organelles which are called spirosomes, which are only seen in endosperm cotyledons of the seeds. Okay. This is in case of plant cells. And the phospholipid biosynthesis is largely done in the membranes of smooth endoplasmic reticulum along with the Golgi complex. And in liver, that is in the hepatocytes. Please mute yourself. So they are abundant and involved in the production of lipoproteins. Then carbohydrate metabolism. So the glycogen is the reserve material in animals, right? The storage food. So this glycogen is first converted, whenever we want energy, first the glycogen will get converted to glucose 1-phosphate, then to glucose 6-phosphate by gluconeogenesis. So this glucose 6-phosphate is then brought to the smooth endoplasmic reticulum. So we don't want glucose 6-phosphate, we want glucose for the immediate energy. So this is brought to smooth endoplasmic reticulum, which is converted to glucose and phosphate by the enzyme glucose 6-phosphatase. So this enzyme is present only in smooth endoplasmic reticulum of animal cell. Now this glucose, which is in the cisternal space, first whatever the glucose produced, that is first transported to cisternal space and then to extracellular space, and then that enters into the bloodstream. So when this glucose enters the bloodstream, the blood glucose levels will be maintained, and then that can be transferred to wherever it is necessary. Okay. And there are other enzymes of carbohydrate metabolism such as amylase, glucuronidase, which are involved in the hydrolysis of starch and mucopolysaccharides. Even they are present in the smooth endoplasmic reticulum. And detoxification of toxic chemicals. So this smooth endoplasmic reticulum detoxifies the toxicity of carcinogens. Okay, so mostly from the food or like some exposure to chemicals, so the carcinogens enters into our body without our notification. So like this, whatever the carcinogens which have entered accidentally, they will be mostly eliminated by this endoplasmic reticulum. Okay, so these toxic chemicals are secreted out of the body by forming vesicles in the smooth endoplasmic reticulum, and it helps in the detoxification of wide variety of xenobiotics like ethanol, barbiturates, aspirin, like drugs, okay, petroleum products, and everything takes place in the liver cells. But when large quantities of harmful compounds enters into the circulation, when when the detoxification fails or accumulation takes place, then they enter into the circulation in the large quantities and in the surface area. So it will slowly detoxify the smooth endoplasmic reticulum. And once these harmful compounds are detoxified and removed, the excess smooth endoplasmic reticulum will be removed by lysosome dependent autophagolysis. So yesterday we have discussed about phagocytosis, pinocytosis, all this. The phagocytosis is a regular process where like excess organelle or cells will be degraded and they will be eliminated. Okay. So in the same way, after complete detoxification and removal of these carcinogens or toxins, there is no role for the smooth endoplasmic reticulum. So that will be removed by this autophagocytosis, okay, which is mediated by lysosomes. So these lysosomes contains digestive enzymes which will digest the components present in the phagocytes. And this detoxification is carried out by a system of oxygen transferring enzymes which are called oxygenases. So these oxygenases, they catalyze number of reactions to make lipid soluble drugs and metabolic waste into water soluble ones so that they can be eliminated in the form of urine or sweat or something like that. Okay. And the enzymes present in the smooth endoplasmic reticulum, they convert lipids and other hydrophobic substances into water soluble so that they can be easily transported through the membrane of the endoplasmic reticulum. So like the important oxygenase enzyme system is cytochrome P450, which is mainly very important and is involved in detoxification of drugs. Okay, and it is iron containing compound having special properties of reducing its form when it bonds with the substrate. So why it got 450 means this reduced form absorbs light at 450 nanometer. And for example, any drug is attached to the cytochrome P450, the iron present in the reduced form, that is ferrous form, the iron which is there in the ferric form will get converted to ferrous form by NADPH dependent cytochrome P450 reductase. So this is the enzyme. Okay. Now this reduced P450 will bind oxygen, and one atom of oxygen is used to oxidize the substrate to convert into from ferric to ferrous. And the other atom of oxygen is used to oxidize the drug with the liberation of one molecule of water. So that means no need to remember the complete mechanism, but just remember that this endoplasmic reticulum is involved in the detoxification of drugs and toxins by employing cytochrome P450. And why it got name? Because it absorbs light at 450 nanometers. Okay, just remember these important points. And other functions of smooth endoplasmic reticulum are, it helps in exchange of materials between nucleoplasm and cytoplasm. And in plant, they establish link between cytoplasm of two cells through a pits or plasmodesmata. So plasmodesmata is like a cell junction, okay, where two cells will attach and there will be a junction where there is a passage from one cell to another cell. So here also, like the cytoplasm between the two cells, plant cells, can be connected through pits. It can form a pit or plasmodesmata. And this smooth endoplasmic reticulum is responsible for the synthesis and repair of membranes. And both smooth endoplasmic reticulum and rough endoplasmic reticulum, they provide area, surface area for the action and storage of enzymes and their products. Now we discuss about rough endoplasmic reticulum. So rough endoplasmic reticulum, so as we have discussed already, it is because of the presence of ribosomes over the surface of the endoplasmic reticulum, which is connected with the production of secretory proteins. So as there are ribosomes here, the proteins will be synthesized. And these are involved in the growth and differentiation because almost, yes.
Blanket
So this is involved in the growth and differentiation. That means the proteins are involved in almost all the process of growth and differentiation. All the metabolic processes require some enzymes, and the enzymes are nothing but proteins. So this is very important. So the protein synthesis is important so that every metabolic pathway takes place. And rough endoplasmic reticulum is named after its rough appearance. Okay, and it lies immediately adjacent to the nucleus, and its membrane is continuous with the outer membrane of the nuclear envelope. And this rough endoplasmic reticulum is found throughout the cell, but the density is higher near the nucleus and Golgi apparatus. Okay, then the ribosomes are, so they are ribonucleoprotein particles present free in the cytoplasm. Okay, so ribosomes can be of two types, which can be free in cytoplasm or which can get attached to the endoplasmic reticulum. So they can either free in cytoplasm or attach it to endoplasmic reticulum or on the outer surface of the nuclear envelope or nuclear membrane. So these are playing important role in the protein synthesis and synthesize structural proteins when free in cytoplasm and secretory proteins when attached to endoplasmic reticulum. That means the type of proteins they are synthesizing is different. When they are present in the cytoplasm, they secrete structural proteins which are involved in the structural aspects of the cell, whereas when they are attached to the rough endoplasmic reticulum or the endoplasmic reticulum, they will secrete the secretory proteins. So functions. First is protein synthesis, transport and folding. So these type of endoplasmic reticulum are prominent in liver cells where the production of proteins is more. And cells are specialized in protein synthesis have more prominent rough endoplasmic reticulum, while smooth endoplasmic reticulum is prominent where the lipid production is more. That means in cells where more protein synthesis is required, there you can see this more rough endoplasmic reticulum. And proteins that are secreted from the cells or act on the integral membrane proteins or proteins of organelle like Golgi complex, lysosomes, endosomes, all these are assembled on ribosomes which are present on the rough endoplasmic reticulum. So the secretory proteins are mainly synthesized and packed and done, everything is done by this rough endoplasmic reticulum. These proteins, instead of passing into cytoplasm, they pass through the cisternae of rough endoplasmic reticulum so that they can escape the action of protease enzymes which are present in the cytoplasm. So when they enter into the cytoplasm, suppose already in the cytoplasm there will be protease enzymes. So that protease enzymes will cleave these proteins. So to escape that mechanism, usually they move through the cisternae of rough endoplasmic reticulum. Then signal sequence. Okay, after packing, where it should go. Okay, so these proteins contain a signal sequence. Like they have 6 to 20 non-polar amino acid residues, which target the nascent polypeptides of the endoplasmic reticulum membrane and leads the growing polypeptide into the ER lumen, that is endoplasmic reticulum lumen. That means it is like an address where this protein has to go, right? So it will have a signal. So signal sequence may vary in length, and it is 13 to 36 amino acids may be length, and especially in that 10 to 15 hydrophobic amino acids, 1 or 2 positively charged amino acids before hydrophobic sequence, and a short sequence of polar amino acids near C terminus, and amino acid with short side chain, that is alanine, must be present in the cleavage site. So this is how it has to look like. Okay, and any amino acid may be present, but 10 to 15 hydrophobic amino acids must be present. So within the hydrophobic amino acids group, anything can come there. Okay. So a signal recognition particle, or SRP, recognizes the signal sequence and acts as a tag that allows this SRP ribosome nascent polypeptide to bind to SRP receptor located on the cytosolic surface of endoplasmic reticulum membrane. That means towards the, you have like endoplasmic reticulum membrane. So towards the cytosolic surface, there will be receptor, which is called SRP receptor. That is signal recognition particle receptor. So that means this particle, what it will do, it will search for all the proteins and it will recognize the signal sequence. And it will go and bind to this signal sequence of the polypeptide. Now we have SRP and nascent polypeptide with signal sequence and also a ribosome. Okay, so this three combination that will go and bind to the cytosolic surface of the endoplasmic reticulum. So this signal recognition particle is a rod-shaped complex. So here you can see in the image. So it has only single subunit like 7S RNA and 6 different proteins. So one protein of SRP directly binds to the signal sequence, inhibiting elongation by strictly blocking the amino acid tRNAs, inhibiting peptidyl transferase. So again, in the molecular biology, we discuss how translation, everything happens. But just remember here that, so whenever the recognition of SRP takes place by SRP receptor, that is signal sequences receptor, so the translation will stop. And as one nascent protein, polypeptide enters this cisternae, it acts upon variety of enzymes located either with the membrane or in the lumen. So then what will do? Nascent protein means, it is always remember that a protein is functional only when it is three-dimensional in structure. So that means the protein has to undergo some modifications and folding should be proper. Then only the protein will function properly. Now here, whenever the nascent protein is synthesized, so that has to be transformed into functional state. And the integral membrane proteins such as glycophorins, and a fraction of erythrocyte, plasma membranes are synthesized on membrane-bound ribosomes and translocated to endoplasmic reticulum as the polypeptides elongate. That means So whatever the proteins which are required for plasma membrane, that is integral membrane proteins or other proteins, so that has to be synthesized in this endoplasmic reticulum. And this integral proteins, if you see by the like look itself, so the outer portion of the membrane, plasma membrane, and inner portion of the plasma membrane are hydrophilic, and the center portion is hydrophobic. So in the same way, the integral protein has to be synthesized and packed. That means the outer stretch should be polar amino acids, and inner one also, and within the channel they should be hydrophobic, right? So this plays a central role in the synthesis and export of proteins and glycoproteins. So I told you glycoproteins means the proteins which are linked to carbohydrates. In secretory cells are best example you can take like liver cells secreting albumin, insulin. That means either proteins or hormones or digestive enzymes, then collagen. So all these secreted by the liver cells. So that is again backed by this endoplasmic reticulum. And these proteins are glycoproteins, membrane proteins, lysosomal proteins, albumins, globulins. So there are many different types of proteins which are secreted and synthesized and secreted by this endoplasmic reticulum. And the ribosomes present on the endoplasmic reticulum are involved in the synthesis of proteins that possess a signal sequence which direct them specifically to endoplasmic reticulum for processing. Okay. So this is how it happens. So these are free ribosomes in the cytosol, and these are the membrane-bound ribosomes. So in the free ribosomes, what happens? So this is the ribosome from where you have mRNA. So to the mRNA, this smaller subunit and larger subunit of ribosomes comes and binds, and they will scan this mRNA. And as they are scanning, tRNA starts adding amino acids, and once there is a stop sequence or stop signal, the translation will stop and the protein will come out, released. Okay, so this protein can go to nucleus or mitochondria or chloroplast or peroxisomes or wherever they have their function. Whereas the membrane bound ribosomes, so here there is a transporter or a channel, okay, which is present here, and whenever the just a second, I will. Sorry. So in the membrane bound ribosomes, so you have like a channel, transmembrane protein in the membrane of that is plasma membrane. So here, this not plasma membrane, sorry. So this is, you have the cytosol here. This is the endoplasmic reticulum membrane. Okay. So in the endoplasmic reticulum membrane, you have the transmembrane protein. And here the ribosomes. So these ribosomes are attached to the endoplasmic reticulum, right? So they will scan this mRNA, and whatever the protein which is synthesized, that will be released into the endoplasmic reticulum, where it will like check for the where this check for the signal sequence, and it will direct the protein to that particular site, whether that protein has to be there in the plasma membrane or secretory vesicle or endosomes or then lysosomes, like that. Okay. So protein synthesized in the rough endoplasmic reticulum has specific final destinations. That means they are very specific, not like randomly distributed. Some may also remain in the endoplasmic reticulum itself, but others may go to the Golgi apparatus, where that will be packed. in the form of endosomes. And they may be directed to lysosomes or to the cell membrane. Or they may also get secreted into the exterior of the cell, that is secretory proteins. So the role of endoplasmic reticulum in protein processing and sorting was first demonstrated by George Pellard, okay, and his colleagues. So how the pathway looks like is this rough endoplasmic reticulum will go to, like, it will synthesize the protein and it will send to Golgi apparatus, where the secretory vesicle will be formed and this protein will be packed and it is sent to the external of the cell. Okay. Then Blobel and Sabatini, they have got Nobel Prize for proposing signal hypothesis. So in 1971. So this mRNA translating secretory proteins or endoplasmic reticular membrane bound with ribosome, that is rough endoplasmic reticular membrane, contains codons at the 3' end, and after initiating codon comes, then immediately one after the other the amino acid will be added by the tRNA and the growing polypeptide chain will take place, which are recognized by this SRP, signal recognition protein, and places the ribosome on endoplasmic reticular membrane for completing the synthesis. So that means how the signaling takes place, that was proposed by these two scientists. Then targeting proteins to endoplasmic reticulum. Now all protein synthesis is initiated in the ribosome, which are present in the cytosol. So till now we have discussed about the protein synthesis on the ribosomes of the, which are present on the endoplasmic reticulum. But now even in the ribosomes which are free in the cytosol, the protein synthesis takes place, and that is destined to the secretion. If that particular protein has to be secreted, then they are targeted to the endoplasmic reticulum by presence of signal sequence. Okay, so it is nothing but like an ID card for the protein. So looking at the signal sequence, the ribosomes will decide like whether this should go as a secretory protein or a protein that has to be involved in some metabolism or something, and according to that, that will be sent. If it is a secretory protein, so then that will be sent to the endoplasmic reticulum. Then that will go to the Golgi apparatus where it will be packed and then it will be released. So whatever we have discussed just now, the signal hypothesis proposed by Blobel and Sabatini, that please mute yourself. That the mRNAs are translated on the membrane-bound ribosomes contain unique set of codons just after initiating codon. So therefore, so it will, the growing polypeptide chain takes place, and based on the signal sequence, so that will be recognized by signal receptor proteins, and then it will be cleaved, and then it will be sent to the, so this is cleaved by signal peptidase. Okay, that means once the protein synthesis takes place, the growing polypeptide chain has to be cleaved and sent to the targeted function where it has to be kept, like either membrane bound or anywhere else. Okay, so this is how it looks like. So you can see here. So in the first step, so this mRNA, so you have signal sequence here. Okay, so this mRNA which is present here, and these are the ribosomes, larger subunit and smaller subunit, and this, it has a SRP receptor in the endoplasmic reticular membrane. So here it is, SRP, signal recognition protein or receptor. So this SRP, if it is present here, signal sequence, so that will come and bind to this receptor. When this SRP and SRP receptors will bind, This ribosomes get attached to the endoplasmic membrane, and this receptor and SRP, which assists this ribosomes to the membrane, will be separated. And then here the growing polypeptide chain takes place, and once this has been done, now a signal peptidase comes and it will clear the signal sequence. So no more signal is required, because signal is only for to know whether it has to be inserted as an integral protein or secretory protein or like that. So once the signal is already read and the process has started, then again there is no use of this signal. Therefore, this signal sequence is cleaved. And once it is cleaved, once the protein synthesis takes place, so this folding of the protein takes place, and this endoplasmic reticulum, it will take this final protein and it will make all the processing. And if required, so as if the protein is a secretory protein, then that will be carried to the Golgi apparatus, right? So this is the step. If you want, I can just read for you. Like, as the signal sequence emerges from the ribosomes, it is recognized by signal recognition particle. And this SRP escorts the complex to endoplasmic reticulum membrane and binds to the SRP receptor. Now this SRP is released and the ribosome binds to the membrane translocation complex, and the signal sequence is inserted in the membrane channel. Now the translation resumes. That means the polypeptide chain starts growing, and then the cleavage of signal sequence by signal peptidase releases the polypeptide chain into the lumen of the endoplasmic reticulum. So this is again a brief steps in the synthesis of secretory proteins. So here, as I told you, like first complex of mRNA ribosome and tRNA complex, then protein synthesis initiates. So same, if signal sequence is there, then SRP binds, it will like directs this SRP, this complex to the SRP receptor, and where in the endoplasmic reticulum, the protein synthesis will be takes in place, and the SRP, which is dissociated from the ribosomes, that will be recycled. Okay, and then finally, once the signal sequence is cleaved, the polypeptide also will be cleaved, and ribosomes get dissociated into large subunit and small subunit. Then this polypeptide is released into the lumen and it will get folded, and the disulfide bonds and other bonds will be formed, and the proteins are glycosylated. That means a carbohydrate group or glycosylate group will be added to the this protein polypeptide chain. So that is for the secretory protein, and this is for the membrane protein. Okay, so where the enzymes involved in the synthesis of phospholipids are themselves, they are the integral proteins which are present in the rough endoplasmic reticulum. And here you have vesicle transport. So once they are synthesized, they have to be transported, right? So there is vesicular transport. So the cisternae of rough endoplasmic reticulum are large cavities which can transport channels of membrane and luminal proteins to be moved from their site of synthesis to the apical tips. So they have to come out. So the synthesis takes place somewhere inside. So they have to come out, and then they have to go to the Golgi complex. Then only they can be packed. And the typical, the apical edges of the rough endoplasmic reticulum are typically smooth. There are no ribosomes. And then there the first transport vesicles, which is leading towards the Golgi complex, will take place. And once these polypeptides are transferred to the Golgi complex, there the package will be done. And how this tertiary structure of proteins is formed. So here, see, whenever the proteins are synthesis, that is only a part of protein synthesis or translation. But proper folding is very, very important. There is any misfolding, that again causes various diseases. Like we have examples of mad cow diseases. So there you can see the misfolding of the proteins. So if a polypeptide is translocated to endoplasmic reticulum from a membrane-bound ribosome, that is called co-translational translocation. Then that means if from a free ribosome, if the protein comes to endoplasmic reticulum, that is post-translational translocation. If from a membrane-bound ribosome it comes to endoplasmic reticulum, then that is co-translational translocation. This is the difference. And the proteins once formed, they folds and forms disulfide bonds by the enzyme disulfide isomerase between two cysteine molecules. Okay, and this requires an oxidizing environment where cytosol provides reducing environment. Okay, so therefore it requires oxidizing environment. So the folding of polypeptide into its correct three-dimensional structure is mediated by chaperones. Chaperones are nothing but heat shock proteins. Okay, they bind to these unfolded polypeptides and stabilize them first to prevent incorrect folding. So once the primary chaperone protein comes and binds, then this is the primary chaperone proteins, that is BIP, binding immunoglobulin protein, which is a heat sensitive protein family, that is HSP70, and this will have a protein binding as well as ATP as domain. So that means this folding is dependent on ATP process. So once the folding is done, so this BIP, it assists in the folding. So it is used to seal the translocon pore when not occupied by the ribosome. Okay, and this initiates the folding. And then the glycosylation takes place. So N-linked protein glycosylation. So there are two types of protein glycosylation. The first type is N-linked glycosylation, which happens in post-translational mechanism. I told you what is co-translational mechanism. And the second is O-linked glycosylation, which we can see in post-translational mechanism. So I hope it is what is the co and post-translational mechanism is clear with you. Now nearly all proteins produced on the membrane-bound ribosomes are glycoproteins. That means a glycosyl group is added to the protein by glycosyl transferase, which is present either in the endoplasmic reticulum or Golgi apparatus. So these glycoproteins are linked to oligopolysaccharides to aspergine residue. So in the protein, wherever the aspergine residue is there, to that this oligosaccharides, nothing but few 2 to 10 sugar molecules together called oligosaccharides. So that particular unit comes and binds to this amino acid. So this is by calnexin molecule this attachment takes place. And so in almost all glycoproteins, a 14 residue core oligosaccharide consisting of 2 acetyl glucosamine, 9 mannose and 3 glucose are added. So this is the composition of the oligosaccharide. And the presence of 1 or more mannose 6-phosphate residues in their oligosaccharide means it is a structural signal. That means that protein has to go as a structural protein. So like that we will have some signal molecules here. So this is glycosylation, which is very important for protein targeting. So here the receptor protein in the membrane of Golgi complex recognizes mannose 6-phosphate signal. If this is present, then the proteins are moved from endoplasmic reticulum to Golgi complex in the transport vesicle. So this will be a membrane bound, not the secretory vesicle. So unfolded protein response. So the proximity of rough endoplasmic reticulum to the cell nuclei gives, so this as they are both together like nearby, so this endoplasmic reticulum can control the protein processing. Okay.
So this rough endoplasmic reticulum can send some signals to the nucleus if there is any problem in the protein synthesis, right? So that can be stopped, again resumed, or can be corrected or like that. So when there is any misfolding or unfolding of the proteins which get accumulated in the endoplasmic reticulum, the signaling will go. So what is that signaling is called? Unfolded protein response. Okay, this signal gets activated and that signal goes to the nucleus. So this reduces the protein synthesis. First it will try to remove the unfolded or misfolded proteins, and again the protein synthesis takes place. And if that cannot be repaired, then the cells are directed to apoptosis, that is programmed cell death. Okay. So here we have, whenever the misfolded protein is there, that will be removed by this ubiquitin complex. So ubiquitin is a conjugated to the misfolded protein which are destined to degradation. Okay. So here ATP is required and the other 3 enzymes are involved here to degrade. So a chain of 5 ubiquitin molecules, they recognize signal for the 26S proteasome. So proteasomes are the vesicles where the proteins will degrade and where the receptors initiate degradation of proteins into smaller peptides and amino acids by the action of proteases or peptidases. So if the ribosomes are there in 2 or 3 as it together, then we can call it as polyribosomes. So the ribosomes are ribonucleoproteins or non-membranous particles. So here they are involved in protein synthesis. They can be either free or endoplasmic reticulum. All this we have discussed. So prokaryotic ribosomes are 70S, that is they have 30S and 50S subunits. And in eukaryotes, they have 80S, that is 40S and 60S subunits. And the 2 subunits are dissociated when there is low magnesium concentration, and they are associated if there is high magnesium concentration. So magnesium concentration dependent association and dissociation takes place. And the smaller subunit fits into the larger subunit. There will be like a groove where it goes and fits. That too only during protein synthesis. And they can form groups. Okay, this contains rRNA and proteins. And sometimes the protein synthesis involves 2 or 3 ribosomes. Then that is called as polysomes or polyribosomes. Okay. So this is how it looks like. 70S, 80S. We have like 50S is the largest subunit. This is in prokaryotes, and 30S in smaller subunit. And this along with this 50S, 23S. So in the 50S we have 23S, 5S rRNAs and 34 proteins. And in 30S we have 16S rRNA and 21 proteins. Whereas in eukaryotes we have 60S and 40S ribosomes, and in the 60S we have 20S and 5.8S and 5S rRNAs and 45 proteins. And in the 40S ribosomes, 18S rRNA and 30 proteins will be present. So these are the composition of these subunits. Okay. So this is the polysomes. That means the translation of a single polypeptide chain can be carried out a pool of ribosomes. So both in the case of free and membrane bound ribosomes, when the ribosomes move some distance from the mRNA, second ribosomes comes and binds. Okay. Again, the third will come. If such kind of assembly is there, then we call them as polyribosomes. Right. So this is all about endoplasmic reticulum. Okay. So today we have discussed about endoplasmic reticulum and the 2 types, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. The smooth endoplasmic reticulum has no ribosomes, whereas rough endoplasmic reticulum has ribosomes. So wherever ribosomes are there, it is mainly involved in the protein synthesis, whereas those in which ribosomes are absent, they are involved in carbohydrate and lipid metabolism. And even though whether the ribosomes are present or absent or any free ribosomes are there, whatever the proteins which are synthesized, that will come to the endoplasmic reticulum. And there will be a signal molecule on the polypeptide or the protein, where by recognizing the signal, this endoplasmic reticulum will direct that protein to the Golgi complex. Suppose if it is secretory protein or if it is membrane bound, so directly that will be transported to the membrane bound, like wherever it is destined by this endoplasmic reticulum. Right? So this is all about endoplasmic reticulum and different ways of synthesizing the proteins. So if you have any doubts, you can ask.
Okay, ma'am. I have a one doubt. Uh, you were telling about the ribosome, na, ma'am.
Yes.
70S and 80S.
Yes.
But there is a number is the difference.
Yeah, it
Okay,
is.
thank you so much,
Yeah.
ma'am. It
Yeah.
is there, ma'am. In 70S, in the
Yeah.
prokaryotic cells,
Yeah.
50 and 30, if we include, it will be 80. But
Yes.
we are counting here 70S only.
Yes, I will clarify. So it is not based on the like addition. So we are not, it is not dependent on like 50 plus 30, it will be 80. So that is not the case here. Here, it is nothing but it is Swedberg's unit. S means Swedberg's unit. So that means how this sedimentation has taken place. So accordingly, they have given the number. That means,
There's difference between, there is a 10 numbers difference.
yes. So that's what. It is not that the number doesn't tell that, okay, 50 plus 30, it should be 80. It is not like that. So it is the why the numbers have given like that is, if you take a ribosome, okay, whether if it is a like whole ribosome, that means small plus large fragment. If you
Okay.
add them in a centrifuge tube, I told you like a gradient centrifugation when I was discussing. So in the gradient please mute yourself. I will explain. Again, you can unmute. So what, like how we separate the molecules, because you cannot see either DNA, protein, or ribosome or endoplasmic. Anything you cannot see with your naked eye. It is only on a gel or in a centrifugation or through a signal or fluorescence, like that only we can determine. So when initially when scientists were like looking for like which one is bigger, which one is smaller, so how many subunits are there, all these things when they're exploring, so they have used gradient centrifugation. So gradient centrifugation is nothing but in a test tube, if you take different concentrations of the sucrose. So for your example, for simple, like for your understanding, I will tell. Suppose you prepare at your house sugar syrup, right? So if you
Okay, ma'am.
add more sugar, it will be thicker. You can prepare another one where you add little less sugar and more water. So it will be less thicker. If you add very one spoon of sugar and if you add one glass of water, it will be very thin, right? So that means
Yes, ma'am.
you have concentration difference here. So when you add here, we are not using any sugars, so sucrose will be added and we will prepare different concentration. And now we add highest concentration at the bottom, then above that we add little less concentration, and above that, like that we add from higher concentration to lower concentration, from bottom to top, right? Is it
Okay,
clear?
ma'am.
Now once you have done that, you can add proteins, mixture of molecules, because all the molecules won't have same density, because the molecular weight differs for all these things. Now here also I will specifically say about ribosomes. So these ribosomes, larger subunit will be more dense, right, than
Yeah,
smaller
ma'am.
subunit. So like that they have added larger subunit and smaller subunit and they have centrifuged. When they have centrifuged, the larger subunit will come to bottom and smaller subunit will be at the top, right?
Okay, ma'am.
Now they have noted the place or point where it has like stopped, means where it has placed. That means the larger subunit will come to the bottom, but not, it won't touch the bottom. So these molecules will always stay where its concentration is matching the sucrose concentration. Are you getting?
Yeah, ma'am.
So the density has to match. There it will place, it will remain stable. Okay. So where it is where it is matching, so they have entered the values. According to scientific this thing, there will be some values. So when they have noted that values, you have like 10 years, 20 years, 30 years, 40 years, 50 years. Say I am giving you example. Suppose at the bottom you have 100 years, then
Yes.
90 years, 80 years, 60 years, like that, till 10 years they have noted. So now when they have centrifuged the smaller and larger subunits of 70S ribosome, they don't know that it is 70S or what. I have taken a bacteria, suppose E. coli. I isolated ribosome. I separated large and small subunit and I added to the sucrose solution. Now I got two subunits. The larger one is, it has stayed at the 50S. It is matching the 50S place. And the
Okay, ma'am.
smaller subunit is, it is there at the 30S place. So I have given, okay, the larger subunit is 50S and smaller subunit is 30S. And now when
Okay,
I
ma'am.
attached both 50S and 30S together, and again I centrifuged. Now it is not that 50S and 30S should be added. So now that has stopped at 70S position.
Okay,
So
ma'am.
I have given 70s. So that means the number is nothing to do with the like addition or something like that. It is
Thank you
purely
so much,
based
ma'am.
on, yeah, based on the concentration density where it has stopped. So similarly to the eukaryotic. Got it? So this is how this was given with a unit. That is S is a
Yeah, ma'am.
Swedberg
Thank you so
unit.
much. 30 minutes.
Any other questions?
No, ma'am.
Okay. So I will share the PPTs in the group. Okay. Don't worry.
Thank,
Maybe by night I will share.
thank you, ma'am.
Okay. Thank you. So we'll continue.
Classes is over, ma'am.
Sorry. Yeah. Today it is over. So
Mm.
I will continue with the next week, like next topics. Okay.
Thank
Okay,
you,
ma'am.
ma'am.
Yeah. Thank you. Okay. So
Thank you,
no
ma'am.
more questions. Right? Yeah. Okay. Thank you so much.
Thank you, ma'am.